Welcome to the last Forever Green Fridays of February. Today we're talking about the Silflower folks. Glad to have them here. I want to start, as always, by acknowledging our lovely funders. Environment and Natural Resources Trust Fund, Minnesota Department of Agriculture, Clean Water, Land and Legacy Amendment, and Builders Vision for Philanthropies, and the McKnight Foundation. Thank you very much. We have some announcements. I know Mary Brackey's going to make a quick announcement first, and then we'll turn it over to Mitch.
Yeah, I'll try to be quick. So it's regarding the summer undergraduate research experience. So this is part of Forever Green's career ways work. And so there will be thank you very quick. There is a summer research experience for students that we're just kind of getting up off of the ground. It'll be sort of a pilot this summer. The aims are really to provide that experience that helps students develop basic sort of knowledge and skills for research, but also to give them a glimpse into the kind of collaborative cross-disciplinary research that is a large part of what Forever Green does to bring about sort of sustainable, diversified landscapes. And as a result of that, we hope that students will be prepared to continue onto graduate research in food and agricultural sciences. So that's basically what we're hoping to accomplish. And what it consists of is students conducting an individualized research project during the summer, working with a mentor. And the program will also involve cohort meetings, so student cohort meetings and enrichment activities. And then they participate in the undergraduate research symposium or expo on campus in August. So for researchers who are interested in working with students, what's important to know is that there is an expectation that you're willing to work with students to put together an individualized research project, and that you'll identify somebody who will serve as that student's mentor. There's also an expectation that that mentor will participate in training, so mentor training. That would be for postdoc, graduate, and lab staff who would be mentoring the students. Okay. And then also that researchers allow students to participate in those sort of educational activities that are a part of the program, which would be about 3 or 4 hours a week. So what do students get? Obviously, they get the benefit of all this experience, skills and knowledge, and they're also paid. Okay, so students will be paid at a rate comparable to $16 an hour for a full-time position for 10 weeks, and that runs June 1st through August 7th. Okay. And so if you're interested in working with a student, what you should do is get in touch with me. I will send you a form to complete with a little bit of information. I'll use that information to promote the research opportunities, working with the different departments on campus, as well as some of our community colleges around the state. Okay. So it's important to note that the program does not provide lodging and meals for students. So students will need to cover that on their own. What else is important to know? I guess, that's sort of the parts of it, and I really encourage people to get in touch with me if you think this is a possibility. Yeah, Louis.
2 questions. One is, when I think about our potential projects, it's really hard to fit them within that time frame. So is that an absolute requirement?
Well, I will say this, that it is a requirement that students are available during that time from June 1st through August 7th. Now, if you would like that student to stay on and continue working, you may do so. But I strongly encourage labs. So people can talk to the undergraduates that they know and encourage them to look at this and apply for it. But that's okay, but I really encourage people to think beyond your own lab. So we want to pull in students from different fields, from different backgrounds, and give them an opportunity to do this kind of research. So does that answer your question, Alice?
So basically, you're drawing from students who aren't necessarily on this campus, which is why
That's right.
the time frame.
Yeah, and to be honest with you, it is behind where I would like things to be at this point.
In the planning.
Yes.
Yeah.
And so for that reason, it might be easier to look at students who are on our campus or in the metro area. But if the student is already doing work in your lab, maybe consider if there are other opportunities, other students that could look.
To broaden the pool of students brought in.
That's right.
Yeah. The other thing is I can't think of any project that would call the student full time. And so I was wondering whether projects within Forever Green could share students.
That's a great question. I love that because ideally, I would like to find or create opportunities for students to work together on research and work with multiple researchers. So this idea of cross-disciplinary would really, I think, be a little bit more tangible and understandable if they were talking with people in different labs and community partners. So I did talk to Emily about opportunities, and I think she had some ideas that could keep students.
For work, it's probably more amenable to this than field work.
Maybe there could be a partnering.
Yeah.
Yeah, Jake.
How many students do you think the program can support? And then, so if it's a limited number of students, I imagine this is like a competitive process. And then what will be the timeline of such a competitive process?
Yeah, so that kind of gets back to the timeline for the rollout of things. It's running short. So I have not heard from a lot of people who are interested in hosting or working with an undergraduate. So right now it's not competitive. So I would say, my hope is we can line things up by have students indicate their interest for this work by the 21st of March.
Okay.
Yeah, just want to echo thanks to Mary and also George and Orr for developing this program and rolling it out. I'll keep my announcements really brief. We had our Senate Ag Committee hearing on Wednesday. It went really well. We had 20 minutes to present. So it was me and then Colin Kiriton and then Peter Frosch from the Greater MSP Partnership, which hosts the Minnesota SAF Hub, Sustainable Aviation Fuel Hub. He spoke about SAF in general, camelina-based SAF in particular, and also our 1 million acre scaling study, which they're a key partner on. So highlighting that work, which is kind of a heads up to the legislature. Hey, this big ambitious roadmap is coming. We're going to need you all to invest in this in a bigger way than you have in the past. And then rounded it out with a farmer, Paul Novotny, who has grown Kernza, has grown roast camelina regularly now, and also started a seed cleaning business for camelina based out of his operation in Chatfield, Minnesota. So awesome hearing, great, tons of really good questions, some pretty effusive comments that really warm your heart. We love Forever Green was stated at least once, I think more than once. So it was great. And the only other thing I'll mention is we are hiring an agronomy associate. If you know anybody who is passionate about doing, a kind of the retail outreach to farmers, going to farms, spending time on the phone with farmers, driving around our beautiful state, and also has an interest in agroforestry and might want to take on some work with hazelnuts and elderberries in that vein, please go to the Forever Green newsletter. All the info's there, and you can just forward it on. We just started announcing it now and are hoping for a good applicant pool.
Just to remind everybody, Silflower is one of the forever green crops that we're working on. It's the target for that crop is to develop a perennial oilseed. It's fairly early compared to other crops in terms of that commercialization path. The domestication of this species really, started around 25 years ago or so with the Land Institute. So, and then we started working with it in partnership with David Van Tassel there. And we have, since that time, developed some breeding populations, explored some agronomic studies, and doing sort of a lot of the groundwork to see if we can get this this crop off the ground. But today we're going to have 4 short presentations of some sort of highlights of things that have been going on in the past year or so. So Renan will start out and talk a little bit about a reference genome. So this is a big development in Silphian, is to have a reference genome as a resource to work with. And then Cesar and Medina will talk a little bit about developing a genotyping platform that exploits the reference genome and other resources that we have. And that's a really important resource that we need to do genetic mapping work and work towards genomic selection, something we've been somewhat struggling with over the past couple of years, but I think we have some interesting progress there to talk about. Then Chase is going to talk about his PhD dissertation work. He's a graduate student in my program, looking at recurrent selection for seedling vigor. And then Sydney will round it out with a couple of highlights relative to the breeding program and a little bit of the agronomic work that we're doing. So,
thank you, Kevin, and everybody for this invitation. It's a great pleasure to talk to you a little bit about Silphian genomics and some of this work we have been doing. So I'm Renan Souza, and I'm a postdoctoral fellow based at Hudson Alpha in Alabama, but I'm funded by the Land Institute. So basically, all this work I'm going to talk, it's funded by the Land Institute in Kansas. So here at Hudson Alpha, I work basically in 2 labs with the Clevenger lab. So in this lab, we focus a lot on developing the actual genotyping strategies for the populations. And the Harkes lab, it's more on the genome assembly and the evolutionary questions about these genomes. And of course, all the materials that we use, they come from David. So David is the mastermind behind all these projects. So talking a little bit about domestication. Just to give an overview, I think everybody here is well familiar with the story of domesticating crops. So if you think about every crop we have today, maize, soybean, rice, all of those, they went through millennia of human intervention, right? So it's basically this very long recurrent selection process that's started for some crops 10,000 years ago, where visually people would select some seeds, make cross sometimes unintended, and evaluate, just by selecting again, and then doing this over and over again. And then as a result, we have, the crops we have today. But again, this is a very, very long process. So, we're talking about a millennia here. But one of the few things here is that, if you think about from almost 30,000 species of plant that could be and have been recorded as usable for human, food or clothing or fuel, only a handful of those plants actually went through this path, right? In fact, if you think about more than 50% of the calories we have come from 4 different crops: rice, soybean, maize, and wheat. But so how can we actually explore, this universe of plants we don't know yet, what they could do for us and what we could do for them as well. Then came this, idea of genomics enabled domestication. So what if we can use all the technology we have to actually sequence and genotype plants so we can find the variants in these genomes that could be associated with genes that are related to those domestication traits, and how can we use those genes to predict and make this process go faster. So basically going from millennia to decades. So a lot of plants are going through this process right now. You're probably familiar with Kernza, Sanfoin, Silflower in this case. So this idea is to accelerate the domestication process of these plants so they are ready to go, so farmers that can use them. So just a little bit about Silphium. So Silphium is this amazing plant native to the US, to the prairies. And it combined two great things. So it's a plant that is a perennial plant. So it produces these very robust root structures that not only help protect the soil, but also make this plant withstand a lot of the stresses, specifically drought stress. And the other important key component here is that Silphium produces these seeds. They have high content of oil, and the oil is very similar to sunflower. So sometimes people like to joke about that is basically like a sunflower 2.0. So, low input, but also highly resilient, that produces these seeds that is also high value for human consumption. So thinking about that idea of, using genomics to domesticate a plant, where can we start, right? So for us, the starting point is actually assembling a genome for Silphium. So here at Hudson Alpha, we actually worked with David to select two different species to assemble the first two genomes for the genus. So the first one is Silphium integrifolium, and the other one Silphium perfoliatum. So those are important species that I use in the domestication program at the Land Institute. And so what you're looking at here is that in the top panel, you have the 7 chromosomes from Silphium integrifolium, so the black horizontal lines. And each one of those panels, you have the landscape in that chromosome. So all the shades of blue, you have repetitive elements. And then at the very top, you have that little scheme of orange, those are your genes. So just by looking at this figure, you can see that those genomes, they are fully saturated with repetitive elements. In fact, about 75% of those genomes are repeats. And similar to perfoliatum at the bottom. So those are, very big genomes, but also very repetitive. And in between those two panels, you can see those gray lines connecting the chromosomes. These represent the synteny between the two genomes. So when you have the lines, ordered like that, it basically shows you that the genomes are very similar in the order or, where the genes are, with the exception of those red lines where you can see, translocations or inversions, specifically on chromosome 1 and chromosome 5. But overall, very synthetic genomes. So Silphium integrifolium and perfoliatum are very similar, but also very big genomes and very repetitive. One of the interesting things we saw, when we were assembling the genomes is that not only these Silphium chromosomes are relatively large, but they present a different structure when they are compacted inside the cell. So I'm going to walk through this with you. So I'm going to try to be as clear as possible, but ask me questions later because I like this story a lot. So what you're looking at here is what we call a high-C map. So it's a chromatin contact map. So looking at this is a chromosome 1 in Silphium integrifolium. So it's almost like shaped like you would expect, a cartoon of a chromosome. So the red signal, it just tells you where a DNA has high contact with its neighbor region. So if you have, a region that is close to each other, you just have like a strong red signal. So naturally, what you would see here is just a single diagonal, just a red line, just showing you, okay, if a region in the genome is close to the other, you just have like this very a strong red signal. But it's not the case for Silphium. So when we look at the chromosomes closely, we actually see this additional diagonal showing up, along the main diagonal. And they have this very conserved distance across the different lines. So what that tells us is that instead of Silphium chromosomes being kind of like in this random spaghetti structure within the cell during the interphase, it actually preserves this almost coiled structure during all cell cycle stages with this conserved 43 megabase turn. So basically, since Silphium chromosomes are so large, they need to preserve additional structure to actually exist in a cell. So this is just one of the interesting things about the Silphium genome that I think it's one of the key features of the Silphium chromosomes. The genome is very, very interesting, but what actually can you do with this, right? So if you're talking about domestication and breeding, we need to use this genome to develop genotyping strategies that are cost effective and also fast and also precise. So one of the strategies we decided to adopt, at least for some of these projects, was target sequencing. So basically, remember that the genome is very repetitive, like I was telling you before. So for breeding purposes and domestication, we don't want to focus on the repetitive elements for now. Let's focus only on the genes because, in theory, that's where all the most important information is for us. So to do target sequencing, you basically design these probes that will bind to the regions that you want to sequence. And this probe, they will capture those regions, so they will select the library. And then you can just put those libraries in the sequencer to kind of like reduce and focus the sequencing only where you want to be, where you want to be sequenced. So basically, we designed this panel we call the Silphium 50K. So it contains about 50,000 sites in the Silphium genome that we want to sequence. And then what you're seeing here in this plot is so the x-axis has about 300 individuals that we sequence, and the y-axis is the number of targets. So it just shows you actually if we do this, how many targets can we recover in the sequence. So we are recovering about 44,000 in the actual when we actually run this assay. So this is actually just showing you, how this data look like. So this is a screenshot from a tool called IGV. So IGV basically allows you to see actually sequencing reads aligned to those genomes. So at the bottom in blue, you see a gene, a Silphium gene, and you have 4 different panels here. Each one of those 4 is a different genotype that we sequence. And you can see that all the sequencing reads, they kind of accumulate exactly where we designed that probe. So just in the second exon of this gene there. So it kind of gives you this control of sequencing specific regions of the genome, specifically when you're talking about big and complex plants like Silphium. Okay, so I'm going to talk to you a little bit about a story, how we use this data to study floral traits, so floral architecture in Silphium. So why we did this, right? So the Silphium capitulum, they have this unique structure that is very different from the other Asteraceae, that in which the seed is actually produced in the ray floret. So there's kind of like this outside ring here, in the capitulums where the seeds are, differently from sunflower. If you look at the sunflower head, all your seeds are in the middle, and then you have the ray florets. But Silphium is the opposite. As you can see here, a mature Silphium head with the mature seeds, but also in the middle, everything, there's nothing there anymore. The ray floret count and the number of seeds in this head is very important for us because it's basically one of the key domestication traits. If you want, increase the number of seeds, it's important for you to know how this is controlled genetically. So we have this collection called the Silphium Association Panel. So David Van Tassel from the Land Institute, he gathered all these individuals across several natural populations in the central US, but also he had some additional breeding material that he developed. So He assembled this collection with 258 individuals, and we phenotyped these plants in 2 locations, here in Alabama and Kansas as well, for 2 years. So both mostly trying to phenotype the capitulum structure. So I'm not going to go through some of these methods behind the GWAS, but I want to show you some of these results. So we have 3 traits here. So ray floret count at the top, seed number per capitulum, and receptacle diameter. So basically the diameter of the base of that capitulum. Running this analysis, we actually found 4 regions that sometimes they occur for different traits in different models and also different locations. So since they were always recurrent, we wanted to take a closer look in these 4 loci that we found that seem to be shaping the architecture of the capitulum. So these 4 loci, each one of them explain about 20% of the variance. And you can see here, what is the effect of changing the alleles for each one of these 4. So for example, if you have the wild type, this is your ray floret count, but if you start, changing that for another allele, you can see, an increase in your floret count, almost like a complete additive effect for these 4 loci. So if you combine these 4 and just create this kind of like a pseudo haplotype of the wild type, you have this GTGG, which would be the wild, so low floret count, and any mutation of any of those components will kind of drive your floret count high. So just to visually understand what's happening, so the GTGG, so this is the pseudo haplotype in those 4 loci. So every time you have this haplotype, you will have a low floret count, like you see in this picture. But if you introduce a change, or if one of those individuals have a mutation in one of those alleles, this is what you get. So you start getting increase in the number of ray florets, increase in number of seeds, and changing the capitulum architecture. We wanted to kind of explore a little bit more to understand, where these alleles they came from and how they actually work together. So I'm going to walk with you here on this panel. So at the top part, you have all those individuals ordered by high floret to low. So it's just like ordering the phenotypes. The second panel, you have the 4 loci, so the 4 QTLs, and also ordered in the same way. And these QTLs, they have different colors here. So everything that is gray is homozygous, so it's low, theoretically low ray floret count. The orange are heterozygous, so they will have at least one different allele in one of those loci. And the red is homozygous mutant, so you have a complete change in the allele call, and then they will, switch to a high floret count. So what you can see comparing this panel and this one is that as you accumulate more mutations on those 4 loci, so as you walk towards the left here, you see that your ray floret count increases, almost at the same rate as you're accumulating those mutations. In the bottom panel, we asked another question about, okay, so how is the frequency of these alleles between breeding material from the Land Institute and wild populations, natural population. And you can see that, so the wild here are the gray, and the blue is everything that is breeding line. So everything that was, a cross or something that, was artificially selected. And you can see that over time, the breeding material, they actually accumulated those alleles. So basically what you can see here is that all the work done at the Land Institute in this last 20 years, it's kind of like the fingerprints of this selection that the breeders they did on the field, accumulating those alleles in breeding material. And you can see this, when you compare basically the ray floret count between breeding material, so anything that was selected, and wild, you can see the difference, in the ray floret count in Silphium. Another question about, these alleles is that we wanted to see, okay, where did these alleles came from? Thinking about natural populations in the United States, right? So they do not come from the same place. So each one of those alleles, they actually come from completely different states. For example, we have some of those coming from Mississippi, Illinois, Oklahoma, Nebraska, Texas. So basically, when you have a domestication program like a TLI, David did all these collections many, many years ago. you kind of like gather all this natural variation and kind of stack them together in single breeding material. So, you provide this, small increments every breeding cycle, but you can see kind of like the story, where all of this came from regarding the natural populations. So talking a little bit about candidate genes. So those 4 loci we saw in that GWAS, they actually, the most significant SNPs are in genes. So, for example, one of that SNP on chromosome 1 is actually on the first exon of this gene, on chromosome 1. So all of those SNPs are actually genic SNPs, but they seem to have not a very strong effect on the protein itself, because all of those SNPs, they are synonymous. So they do not, it's not possible at the moment to establish a causality. Like they do not cause the mutation, but they are probably linked to the mutation, as far as we know right now. So we know where the SNPs are in the genes, and also like we know what those genes they are possibly involved in, which functions they're involved in the silphium structure, but it's hard to tell right now what exactly is the effect of those SNPs in the protein that they are, possibly producing. So that's where we are right now with this study of the floral architecture. But I just want you guys to remember a few key points here. So basically, Silphium is this amazing system with great potential to become a new climate resilient crop, and we have been using target sequencing for these initial studies as one of the options for sequencing. And then these new candidate genes are, we're thinking about developing markers to actually start using that to screen breeding material, the TLI. And I also want to say, because that all this data is available, I finally finished uploading all of these data sets to NCBI. So if people are interested in taking a look, using data sets, those are the project numbers. You can just go there and download everything. And we just put a preprint out about the Silphium genome early this month. So yeah, please go take a look as well. We, would appreciate any feedback you have about this project.
First of all, awesome work. The floret count and seed number work, is that just kind of the first test case to try to, draw useful, breeding useful information out of the assembly that you did? And is there a plan for, future target traits to look at?
Yes, yeah, that's exactly the case. Yeah, because we actually we collected more phenotypes on this panel. So we have some of disease resistant work, a little bit some of the other morphology traits, plant height. But this was kind of like the first story we wanted to put in this first publication. But we do have a few follow-up analysis we want to combine in, following other studies about this GWAS panel.
Did you consider doing RNA-seq?
To test those
for genotyping? RNA-seq for genotyping? Was that just like the cost was cheaper to do?
No, yeah, we there are other folks that worked on using transcriptome assembly in Silphium to kind of like, this idea of reducing representation for the genome. The problem though, you can get, if you focus on the transcriptome, you get, reduce the representation, but it's still very expensive to do this in a large scale. So we just wanted to do something very, more affordable using target-seq. But yeah, there are other people that tried, focus on the transcriptome, but it's I would say to apply that for a large scale breeding operation would be a little bit challenging due to the costs.
This is a continuation of the Silphium project, and as Renan introduced before, Renan has been working in the Land Institute, and this part of the project has been developed here with Kevin Smith data and all related with the University of Minnesota. Of course, the Renan works impressive, and they have a lot of more resources and more time working in Silphium than me. But still, I will show a little bit about the molecular breeding here with Silphium population. And of course, something that we want to do is to improve our genotyping platforms, but always try to keep up the cost lower. Genotyping is expensive and Silflower is huge, so the idea is to find the correct threshold, the correct balance between those two issues. I have been working with Kevin too in a Silflower GBS project who has been developed during several years, and now I am trying to just organize and clean and obtain good results from that project. And in general, this GBS project is a panel of 360 plants genotype that were genotyped with GBS. And the idea behind that was, okay, we have this information, genotypic information with GBS, and also we have a phenotypic data collected across multiple years. We will try to identify molecular markers and try to train a good training population to run genomic selection or genomic prediction first and genomic selection later with Silflower. In this case, I start to work with this Silflower GBS project. And the first step was to, using the FASTQ files from GBS, try to generate a high quality BCF file and compare the imputation methods, try to identify the best way to impute our BCF files, and also identify other characteristics that we can find in this population. For example, population structure, linkage disequilibrium, yes. And then with that in mind, we can continue with the next step. That is the run the GWAS, run the genomic selection, and try to find something in this panel. This table represents the data set that we have in this moment here in with Kevin group. And in the first row, we have a big information with 19 genotypes that were genotyped with whole genome sequencing. We have a lot of sites, around 150 million of SNPs of high quality, because those 19 genotypes were sequenced with whole genome sequencing, keeping a high deep sequencing. And the next row, yes, correspond to the BCF files generated from GBS data. In that case, we have more individuals. The big panel contains more than 1,100 genotypes, plants, and the number of sites or number of markers initially is really high, but like Silflower is huge. We have a very sparse data, matrix data, but after we start to filter by quality and maximum missing places, we reduce our number of sites to 158,000 SNPs. That is a huge reduction, but that is because we are filtering by quality, by use sites that have a lot of genotypes in common, and then we start with the process of imputation. So just to remember, we have several groups, several number of genotypes. The big one, yes, the project 20 contains more than the 1,100 genotypes. Into the 1,100, we have a subset of the population of interest, the purple one, that this population of interest contains genotypes, plants with GBS, yes, and also phenotypic data. And in the green one is a subset of 19 plants that were also genotyped with GBS, with whole genome sequencing. So that is our subset. But also when we identify the Venn diagram between the whole genome sequencing with 100 million of SNPs versus the BCF file from GBS data, we identify 22,000 markers in common. That is not a big number if you see, because I was expecting to have all the data from GBS into the whole genome sequencing, but that is not happening. And the reason is because we are just subsetting a small number of genotypes, 19 genotypes. With that in mind, we start a procedure, one evaluation of the different methodologies of imputation to identify the best way, yes, the most accurate imputation method. In this case, we were using our 19 genotypes with whole genome sequencing to validate our GBS data. In this case, in general, we will assume that our ground truth BCF file is the whole genome sequencing. Yes, and we are trying to impute with different methodologies, and then we can compare, yes, the imputation versus the reality. Of course, we can identify the concordance, also the precision or positive predicted values, and the sensitivity of the different methodologies. I was comparing two different methodologies. One is Beagle, that is one single imputation method. Another is GLIMS. GLIMS is using the 19 whole genome sequencing genotypes as information, as prior to do the imputation. Beagle is not using anything, it's just impute. To run this analysis, here I was interested to compare Beagle versus GLIMS. We will focus in these two, and we identify that Beagle in these two, and we identify that Beagle, yes, concordance, accuracy and sensitivity in different versus GLIMS. GLIMS always keep higher values, even really high values in sensitivity and concordance in comparison with Beagle. But the problem is that GLIMS also impute SNPs have in common with the whole genome sequence information, right? That is the reason, because the number of SNPs with GLIMS is only 9,000 markers versus 25,000 markers from Beagle. Finally, we repeat the procedure with this was done with the total number of genotypes from the project 20, the 1100 genotypes, but we repeat the same process with the panel of interest, these 360 genotypes that were genotype and phenotype, finding similar results. So we keep, in general, we identify that we can use even the small panel with 360 genotypes or the big panel with 1100 genotypes. Then we identify what is the population structure of this panel. In general, we identify a population structure. The upper panel corresponds to the principal component analysis of the total individuals, 1100, and the small one corresponds to the principal component analysis of the 360 genotypes. Identified by families, that is one of the most interesting parts. The families are shaping the population structure, and also the linkage disequilibrium is larger when we are using just the small subset of panel, the 360 genotypes, right, in comparison with the total 1100 genotypes. The reason is because in the 1100 we have a lot of diversity from multiple sources, and the decay is really fast in this case. We also compare the results in general, GLIMS versus Beagle imputation methods, and there is not a big change in the population structure. This is quite similar. And also the marker distribution is really important. We have a subset of markers in the upper figure. In Beagle we have 25,000 markers. In the lower part we have 9,000 markers, but the distribution is evenly spaced. Yes, in both cases, it's difficult to find equal, yes, space, and even it's more difficult when we are working with a huge genome in this case. But I think that in both cases, the methodologies are fine, but we will continue with GLIMS because the accuracy of imputation was higher in this methodology. Then we identified how many markers, yes, we have separated by a distance greater than the linkage disequilibrium decay. We have a linkage disequilibrium decay of 4.3 megabases calculated for this population. And we identify that in total we have a 593 markers that were separated with a distance greater than that. And that is fine, because that only represents 6.5% of the markers. So only 6.5% of markers are separated, have a gap larger, yes, than the linkage disequilibrium decay. The other markers are fine, are evenly with a short distance. And with that in mind, checking everything, we continue with the next step, that is GWAS and genomic selection. For that part, we identify, we start to work with in total 22 different traits collected from 2020 to 2025. They are agronomic traits related with flower, stem, biomass, leaf. The idea was to model the phenotypic data to obtain bloom values, and then use those blue values to run GWAS and genomic selection. Some traits where most of the traits were evaluated multiple times. So this is repeated, they were considered as repeated measurements because the traits were collected as individual plants. And also the genotypic information was collected as individual plants. So, but the red boxes correspond to traits evaluated once, only 1 year, but most of the traits were collected multiple years. So it was easy to model those kind of traits to obtain one single blue value. And after run the GWAS using ASRemel, we identified in total 51 markers in 18 of 22 different traits located across all different chromosomes except for the chromosome number 6. And additionally, we identified, for example, one marker in the chromosome 7 that is associated with 4 different traits. We identified 2 markers in the chromosome 1 and 3 associated with 3 different traits, and so on. Some parts we can identify some regions that are quite close. Yes, markers that could be close, but in this context, close is relative because close is around 4 megabases, 5 megabases. Looks close in some parts, but they are not really close. In general, for example, I have this example of one marker associated with seed head number of seeds, with a high phenotypic variant, phenotypic variant explained. And also we can see that there is an increase of 13 units in this heterozygous state versus the homozygous state of the mean value. So this is an interesting marker. And the next step for this GWAS is to run the annotation, as Renan showed before, and identify linkage disequilibrium blocks. Additionally, I run the GBlock to identify also the genomic prediction values. And in this case, I was calculating the predicted ability, the column PA in the table. And we identified that 12 traits contain predicted abilities higher than 50%. And we identified, for example, flower date by individuals with high predicted ability of 77%. And also for harvest date. Something interesting that we identified was the following. We can plot here the coefficient of variance of the blue values that we already modeled across multiple years. And also we identified the predictability. And something that happened is that when the coefficient of variance is pretty low, that is something that we want after modeling, of course, the predictability is really high. Something that is happening here in these dots that are here in the upper part and the lower part are the data, the traits that were not modeled because they were just collected once. So that is what is happening. But in general, we have a good, a good correlation, negative correlation, for this model, and good predicted abilities in general. The next step for this one, if we want to improve the predicted abilities, is to incorporate the molecular markers that we already associated from GWAS in the genomic prediction model. That is all that always increase the predictability for the model. The next step is something related with our genotyping platform that we want to develop. Something that will continue with Renan project is to develop a genotyping platform to genotype multiple populations, in the future as cheap as we can get, but also with a high accuracy, good value of each marker, and also that are consistent across populations. Like I was working with alfalfa have been working with this dark tag approach. In total, now we have more than 16,000 genotypes with data approach. And in this, this is a good scenario to repeat the same process with Silflower. Our idea is to use the information that we have here from the whole genome sequencing, GBS data of all the markers, the high quality markers from GBS data, and the marker that we're interested from different traits to be used to develop our dark tag panel. The panel in total will contain 3,000 markers. That is a very few number of markers. If you see 3,000 markers, it's a small number, but our question was to know if that number of markers was enough to keep, to be informative for our proposals. In this case, we run genomic prediction using 10 random subsets of 3,000 markers, 6,000 markers, or 9,000 markers from our BCF file. And we evaluated again the predictability of the flower date traits, and we evaluate how was changing the predictability, right? In this case, we identified that the reduction was from 1 unit when we reduced from 9,000 markers to 6,000 and to 3,000. But still, this is valid. It's something that we need to try to identify. Yes, the best way to keep the most informative markers, yes, with the cheapest way, and also keeping our high predictabilities. Could be great to do this, to develop this platform, because in any case, DARTAG is consistent. The same 3,000 marker will be consistent independently of the population that we are genotyping, something that is not happening with GBS.
Is the price to go from 3,000 to 6,000 markers double the price?
Yes.
Okay.
Yeah, that is something that they just develop another array and the price stop.
Okay.
Maybe a little bit lower because you only need to extract the DNA once,
Mhm.
but the big chunk is higher. In alfalfa, we are paying $15 per sample, so it's fine, including the DNA extraction. In conclusion, we identified something really nice for our population. We identify our population structure, linkage disequilibrium decay. We identify a low number of markers separated with larger distance than the linkage disequilibrium. Also, we identify our blue values that were useful to identify multiple molecular markers in most of the traits. And additionally, we identify high predictabilities for most of the traits also. And also, we identify that using 3,000 markers from this BCF file, we can keep highest predictabilities as the 9,000 markers from the GBS, from the original BCF file. That is fine because our next step is to develop or synthesize the oligos for DARTAG and implement the DARTAG approach in Silflower. Try to change from GBS to DARTAG. But in this time, the idea is to finish this project because it's very useful to finish to use the data that we already have.
These two projects have been really interesting because we've been exploring multiple different paths to try to come up with practical tools to help the breeding program. And I think that these two approaches will be quite complementary to one another. So the approach that Cesar just talked about is really, the discovery panel is really more the germplasm that we're working with. What Renan was talking about was developing a very, very diverse set of germplasm in that discovery panel. And I think that platform will be quite useful for, bigger diversity studies or genetic studies. So in the end, I think they'll be complementary, but, it's been sort of a challenge to think about all of the issues of how to come up with a technology that we can use, at least for the near future, to kind of advance the genetics of the breeding work. So,
Those are pretty good predictabilities.
what's that? Yeah, the predictabilities are as good as my barley program, so I'm not going to complain at all. Yeah.
The project I'm working on is the Seedling Vigor Recurrent Selection Project. Silphium in the field has large challenges with stand establishment and seedling vigor. So we see skips in the row, poor germination, and that leads us as a breeding program to utilize transplants to do all of our plots. Of course, this is not a commercially viable way for farmers to plant this crop in the future. So what we're looking at is to develop populations that have high seedling vigor that can be directly sown into the field. So this project comprises about 72 families, selected both from UMN material and the Land Institute material. And the strategies that we're using are two selection strategies. One is called Best Overall, where out of 720 individuals, we pick the top 72, regardless of their family. And those are then intermated within a pollination tent. Bumblebees are introduced to pollinate, and they're intermated with one another. For best individual, what we're doing is, out of 72 families, we track the maternal line, and we grow them out, 720 individuals. We pick the top individual for the total leaf area. That's the only trait that we're selecting upon within this selection scheme. And we're picking that one individual from each family. Those are then intermated. And then for random, there's just a random selection out of the 720 individuals in that population, and that is acting as our control to determine the effective selection over time. So this is the 4-year process. We're now in the final year of creating these populations. Currently, we are vernalizing the plants in the coolers. They'll come out, they'll flower in May, like early June. And then that seed will be used for a field experiment in the future. And we started about 5,400 individuals in cycle 0. So for the field experiment, the purpose of the field experiment is to determine if genetic gain has occurred. So we have different seed lots. So once the plants have flowered in the greenhouse, they're planted out into the field, isolation plots that are far enough apart that they won't have enough pollination between one another. And these seed lots are collected every year. So for cycle 0, by 2027, we'll have 4 seed lots. For cycle 1, we'll have 3, etc. That field experiment will then be conducted for 3 locations: St. Paul, Rosemont, and Becker. And then we're going to take the same traits that we take in the greenhouse in the field. So that's germination rate, emergence state, leaf number, leaf width, etc. And the most important one is total leaf area. So here are the selection differentials between cycle 1 and cycle 2. So as you can see, best overall for both cycles has a pretty large selection differential. Best individual has a decent amount, and then random, as expected, has very little selection differential. What this tells us is that we have not exhausted the variation for seedling vigor or total leaf area within the population as of yet.
So total leaf area is your measure of vigor.
Yes, it's a proxy trait of vigor.
Gotcha.
Yeah. And then here again, we have cycle 2 to cycle 3. Cycle 3 was just measured in January. And those, again, we have a large selection differential between best overall and best individual, and not much for random. This is a chart determining maternal pedigree across the different cycles. So on the y-axis, we have the maternal families of all the 72 families that were introduced in cycle 0. And then as you go from cycle 0, cycle 1, cycle 2, and cycle 3, you're losing families as you select the top individuals from each group or within the group. So we have in the highlighted red section are 2 families, and those are both from a population called the UMN inbreeding program or population. And those contribute about 53% of the contribution within cycle 3. So that's really interesting. And you can see that the red line indicates what is UMN material above the red line and TLI material down below. We have selected, it looks like, for UMN material. And there's only one family that has consistently stayed from cycle 0 to cycle 3 that's from TLI material. So that's really interesting to see how we're selecting for these type of maternal pedigrees. We don't know the paternal lines. We'll know that when we find a marker assay that works for us. We'll genotype everything in these panels or with each cycle of selection. Here is random. So here you can see that TLI human material have been selected relatively randomly. There are some families that are increasing due to genetic drift. We'll know if that is true when we do the marker data as well. If you look at the same 2 populations that we see of high amounts contributed in the best overall population, they actually are pretty low in the random population, so which is to be expected. This is just the percentage of remaining families within each group. So best individual has kept about 58 families, 81% of the original family maternal lines. Best overall has the highest reduction at 18% or 13 families. And then random at 31%, 22 families represented. We'll take these populations, they've had tissue collected, and we'll use the new marker array that's been developed, and we'll be able to track the accumulation of favorable alleles for total leaf area and other traits that we've collected over the past 4 years to determine what types of SNPs that might be useful for a breeding program to select for total leaf area or predict total leaf area or seedling area.
Is the goal of having that best individual group just to maintain more diversity?
Yeah, so the goal of best individual is to determine, Silphium is self-incompatible outcrossing species. If you bottleneck too quickly, we've seen inbreeding in John Hill Price's previous work of inbreeding depression. So the two selection methods is to determine if we bottleneck too quickly, are we losing favorable alleles? And best individual maintains that variance and still selects for total leaf variant. Another side project that I'm working on is Silphium tissue culture. Currently, we are using for clonal division, crown division, in order to produce clones for different experiments or different types of research or ornamental use. There's a need to produce a lot amount of clones. Right now, we're digging crowns in the field, potting them up in the greenhouse. It's very labor intensive. It's not very scalable. So I built off a sterilization protocol that a previous grad student did for Silphium. Silphium has a lot of endophytes within its tissue, so it's very prone to contamination within the tube or on the auger. So I modified that sterilization protocol and have began testing different varieties of auxins and cytokinins to produce shoots and roots. We've seen promising results for at least the cytokinins. They've produced shoots. So on the right here, you see the axil buds. Those were dormant, and then when placed on media, they've since sprouted. So that's really exciting. And if I'm able to get both roots and shoots, then we'll be able to produce a lot of clonal propagates from our material. And then another fun thing we're doing is we talked about ligule counts. So there are some genotypes within this UMN breeding program that has 100 plus petals or ray florets that are of ornamental value or could have potential to be ornamental within the landscape. So we've been working with UMN Horticultural Trial and Display Garden here at St. Paul campus to display ornamental genotypes and determine if they would be useful for the landscape. And this is also a great opportunity to provide signage opportunities for the public to engage with research here at the university. And go from there.
How satisfied are you with the progress? You clearly are making improvements in that leaf area trait. Do you feel like you're getting to a point where you've got bigger seeds, or is it just, we're moving in the right direction?
They're definitely, anecdotally, we won't know until the field experiment growing faster. They seem to be really easy to grow, the higher germination rates. But it'll be interesting when we do a pilot field experiment this spring if that is seen in the field as well.
This is a really terrible thesis experiment because it takes 4 years to actually find out if it works. But good for Chase to take that out.
Well, and we've even seen some premature bolting
Yeah.
in the best overall groups, meaning that there's just kind of general bigger overall. It's not just
They're
necessarily.
reaching maturity sooner, and then they're bolting in the greenhouse without fertilization, which is nice for speed breeding if that ever. That's the thing.
Yeah.
I just have two questions it's mainly for curiosity. The first one is about the simple component analysis that we use. PCR, in situ. And the one to know your opinion where you use this simple functional analysis, the sum of the two axes should be a little bit higher, but from your side, your slide, I just something a little bit less, but your prediction is very high. I just want you to comment on that. And the other one is you use leaf area as a proxy for bigger, and I'm not really 100% sure why you use only leaf area as a sign of bigger, because in some publication I've seen also the biomass after below the ground, it affects the biggerity of the seedlings.
Yeah, so the reason that we're using an above ground total leaf area is that it's non-destructive, it's easy to measure, and we've seen in other crops like cured clover, they've done total leaf area before, and it seems to work for them. So that was pretty much the proxy trait that we're really interested in. And then PCA is.
Yeah, PCA is.
So did you hear that?
Oh,
Question was
Cesar.
for you.
Cesar,
But,
yeah, there was a question for you.
Yeah, there's a question.
Cesar has a question.
Yes.
Let's come back to that question and we'll let,
Okay.
Sydney.
I will focus on that.
Yeah, I'm here to provide an update on breeding and field methods. So just give you an idea of the fields that we have currently in Minnesota within the breeding program. We typically have a crossing block every year here in St. Paul. And then we have yield trials at Kirkston, Becker, Rosemount. And then we usually, at least in the past, any advanced lines that we've taken from the yield trials, we'll put in isolation plots and then develop synthetic populations from those. But we are thinking about changing the breeding scheme around. Well, we'll see. We're going to try it out this year, but so we'll see what happens. This is kind of the breeding scheme we've developed, and like things kind of flow from one into the other and to the next, etc. Great. Just to give you an idea of what our preliminary yield trials look like, these families are selected from open pollinated parents with either seed or forage characteristics. The seed lines we select based on high seed yield, early flowering time, high thousand seed weight, and short kind of stature. The forage lines we have selected based on high biomass yields, late flowering time, and more of a tall stature. These experiments consist of 11 to 20 families and then 2 to 3 replicates within an experiment. And this results in about 33 to 40 plots total with 10 plants per plot. And this is what the plot structure looks like. This is actually the crossing block we have here on campus, but this is a good enough proxy. You can actually see, I don't know if this works. No, it doesn't. So in that last plot there, it's kind of fun. There's a couple of plants that have aster yellow, and you can see that they stick out because they're a little more yellow. And this is satellite imagery taken using Google Earth. So there's some exciting things on that end, but I'm not going to talk about those at all. This is just for you to get an idea of what our yield plots look like. So one thing that we're looking at is changes in traits with cycles of selection. Like I said, our preliminary yield trials kind of in that structure like I was talking about before. In the first year, we usually measure flowering date, height, number of stems, biomass yield, seed yield, and then 1,000 seed weight. In this case, I have 4 different cycles that I can show you today. Ones that include our 2019 preliminary yield trial in Crookston Rosemount, and then our 2021 through 2023 preliminary yield trial data from Becker, Crookston, and Rosemount. And then today I'll just show you kind of how things have changed with these cycles of selection for flowering time, seed yield, and 1,000 seed weight. And keep in mind, these traits are average over all families within a single cycle and within a location. So this graph on the x-axis, we have seed yield, 1,000 seed weight, and flowering time, days after May 31st. And then on the x-axis, we have a location, so Crookston, Rosemount, and Becker. And as you can see with seed yield, we have a nice little increase over cycle selection at Crookston, but then we see this kind of decrease, at least initial increase with the third cycle of selection in Rosemount, and then kind of a continual decrease. I believe this is due to the fact we've selected a lot of lines out of Crookston. And so I think we've selected for plants that yield well in Crookston. And we haven't necessarily considered plants that yield well in Rosemount or Becker. So we'll definitely have to consider more of those traits in the future. As for 1,000 seed weight, kind of a similar trend. We had like a little bit of an increase for a few cycles at most locations, but then we've kind of seen this decrease with the current cycles of selection. And once again, a very large increase in Crookston. And I think too, like one of the big seed traits that we select on is seed weight in Crookston, because those plants typically have the largest seed of any of our locations. And then for flowering time, what we'd like to see is a decreasing trend, right? Earlier flowering time is better for us because that means earlier harvest time. And with the short seasons here in Minnesota, we want to try to get that seed out as soon as possible. And we do kind of see that trend, at least from the first to third cycles in Crookston and Rosemount and Becker, but then we're kind of seeing this uptick again in flowering time. And I think that's because in cycle 5, we've selected for more forage types. So I think those types are kind of clouding what's going on. And in the future, I'll definitely have to make like a
cycle of selection versus method or type of selection, to kind of point out what's happening in our forage lines versus what's happening in our seed lines. Great.
That could explain more than one of those traits, right?
Yes, yeah. And most of
Contribute to.
Yeah. Well, and typically too, with higher forage biomass, we do see higher seed yield.
Oh,
So, yeah, which is, yeah, but we also see later flowering time, and that's not necessarily something we want, and taller stature. So we're also looking at how traits change with age. So Cesar talked about this population a little bit in his presentation, but I'll talk about the 2019 preliminary yield trial in Rosemount, Minnesota. The field was planted in 2019 and didn't flower for the first time until 2020. Silphium needs a full year in the ground before it will flower and needs to fertilize before flowering. These individuals are from 11 open pollinated families. And in 2020, we measured traits on the plot level, including seed yield and 1,000 seed weight, but then individually, flowering time, stem height, leaf thickness, and stem thickness have been measured. And then from 2021, basically when I got here, to last year, we've just been measuring these traits on an individual level to keep things a little more consistent. So we can look at how these traits vary in individuals from year to year and how that's changed within a family as well. So the big question I guess we have with this data is, how do yield and physiology traits change with age in Silflower? This is only 11 families at one location, so it's hard, but we also do have experiments where we have families that were planted in different years, so we can look at the effect of age and location and site year in those cases, but maybe not necessarily these. But this is the most data we have. We have 6 years of data from this population, so we'll see what it looks like. So just to get an idea of what these yield traits look like over time, on the top left panel, we have seed yield. And I should clarify, each of the different colors is a different family within these different, or between these 11 different families. And we clearly have some families that yield really well, even at age 4, which is very impressive, but we still do see that kind of yield decline over time. One caveat, we are measuring individual seed yield, but we're not collecting all of the seed because that would be crazy. We are collecting a subset of the plants, so a 3-stem subset, and then counting the number of stems and getting a 1-stem estimate, multiplying that by the number of stems, and that's where we're getting our seed yield metrics from. So I think these seed yields are a little inflated because of that. But I do still think they're informative and will help us in selecting families that yield well. All right, in the next panel, we have 1,000 seed weight, and we can see there's kind of an up and down trend. What we expect to see is a declining trend over time. And I think we do see that until about year 4. But then, I don't know, I think the environmental characteristics kind of come in at that point where, like, if you have a year with increased precipitation or more water availability or more nutrient availability, you may get larger seeds versus more of this like continually declining trend like we would see otherwise. Flowering time on the bottom left panel is pretty consistent. You can see it's dependent more on the year than on the families themselves. The families are pretty consistent in terms of flowering date. And then for stem height, we do see a slight increase over time. And this again doesn't look like a family. In fact, I would say it looks pretty continuous across most of the families. And then these are just little regression lines plotted over those points. Once it said, like I said, kind of an increase in seed yield. But I think looking at different types of patterns and trends will be more informative than just looking at a linear regression. But this just kind of gives us an idea of what's going on. And then kind of a general negative trend with 1,000 seed weight. Kind of a static trend. It depends on the family, of course, with flowering time. It doesn't really look like there's a slope, but there are certain lines, certain families that are more or less affected. And then stem height, we see a general positive trend over time. And then we'll also look at this for plant physiology traits, including top of stem diameter on the top panel and leaf thickness in the bottom panel. And what we'd expect to see for the stem diameter is to decrease over time as silphium grows. It grows more stems. As it ages, like it'll typically start with 3 stems, and the next year you'll have 6 stems, and 12, etc. And with those stems comes increased competition, and with increased competition comes decreased stem diameter. But I think instead what we're seeing is kind of, once again, at year 3 or 4, this leveling out in terms of stem production. and therefore kind of a standardization or like a kind of maintenance, if not like a small increase over time. And this might be too, I've got to see when they applied nitrogen to fields and whatnot, because that definitely affects plant growth. Right? Okay, but what's kind of interesting, leaf thickness is we see a general increase over age. So the leaves are getting thicker as the plants are getting older. Whether this is like more of a perennial growth trait, or is the carbon becoming more stable over time? I don't know. I'll have to look into more papers to state what's really going on there. And we'll look at regression lines for these again. As we can see, certain families are definitely more affected by the decrease in stem diameter than others, but most families experience an increase in the leaf thickness with age. All right, like Chase was talking about, so I'll switch now to silphium establishment. Currently, we use transplants in the field. However, we would love to direct seed. Silphium only produces a rosette during its first year of growth. And it's highly susceptible to weed pressure during the rosette year of growth. So you can see here on the right, there's this nice little 5-row plot or something of silphium that I was able to help establish at Kirk Kimber's farm. We did plant silphium to the left of it as well, but I wasn't able to weed it, so it all died. So we suffer a lot from weed competition out in the field. However, this last year we did this barley interseeding and herbicide trial where we applied a pre-emergent herbicide on the plots, and it's kind of hard to see, but that patch there in the middle on the left with the bare dirt is where I applied the pre-emergent herbicide, and the horrible mess before that is where I did not apply pre-emergent herbicide. So it definitely had an effect on weed suppression within silphium fields, and I think we'll have to integrate this into our production plans in the future. And then the picture on the right shows the barley actually intercropped with silphium, and the silphium growing pretty well under it. But I think you can kind of see still in the background that mess of weeds where we didn't apply a pre-emergent herbicide. So it's definitely mandatory, I think, for silphium establishment. And we planted another trial last fall that we're going to do similar treatments, look at multiple different pre-emergent herbicides this year. And for the first time, we actually used the seeder to plant silphium out in the field. So I'll be excited to see if it even comes up. We might have planted it to do, but we'll see. And then Grace Doyle, who is also here, is also performing a seeding trial using, you can kind of see the tractor there in the background, the Truax planter, into 2 different types of media, either tilled areas or areas left with stubble, and then 2 different planting dates within those. So we'll see how the silphium performs within those as well. And then harvesting update 2025, I remembered in 2023, we had, or 2024, we had TLI come up with the flailback harvest. We've harvested the combine in the past, and now we have a stripper header. Yay. The mechanism of the stripper header, it has these fingers that ideally take the heads, scrape off the seed, and then leave the sticks behind. And then within the mechanism that we have, it has a belt in it that takes the material to like a nicely placed bucket on the side. So we can, oh yeah. And then this is the stripper header that we received in June of 2025. So this is a video of Guillermo using the stripper header last year. Maybe it's a little low, but this is a sample. You can kind of see the sample filtering into the bucket. This is a sample from another plot that we got. And what we're hoping for is more leaf and seed material and less stick material. And that's what we got in some cases. So that was really nice, but not necessarily in all cases. So there are some limitations with the stripper header. It has these really nice bars on it that like to push over the plant material before the header can get to it. So that's a modification we'll be making this year. And then the hoses themselves on the stripper header are very long, or actually very short. So we're going to extend those hoses so that they're able to reach kind of our taller plant material. But then we also have limitations with the plant material we have. The greener heads in the front are pretty much the same age as the brown heads in the back. So if we can increase like maturity timing within silphium, and then also decrease kind of the flower distance to where things are more consistent, because the stripper header, if things aren't kind of within this window, it's just going to start chopping off sticks. And we don't want sticks in our samples. We just want seeds and leaves. So that's just something that we have to work on, kind of breeding stuff.